Hello again. Now, before we dive into the chemistry, let's make one thing clear from the jump. Everything you're about to hear is strictly educational. This isn't advice, it isn't promotion, and it isn't encouragement to use, grow, buy, or do anything in any place where it isn't legal. What we do here at Canna Intel 420 is break down the science so people can understand the plant, the chemistry, and the biology behind it. That's it. So, with that out of the way, let's get into this properly. Durban Poison. A name that gets tossed around everywhere, usually with more mythology than chemistry behind it. But today, we're not dealing with stories. We're dealing with the real molecular architecture that defines this lineage, what the plant actually builds, how the body actually interacts with it, and why its natural chemistry behaves differently from the stripped-down, lab-assembled versions people often confuse it with. If you're here for hype, this isn't that. If you're here for the truth, you're in exactly the right place. I'm Jerry, and in the next stretch, we're going to unpack this thing layer by layer. Cannabinoids, dominant monoterpenes, receptor-level interactions, digestive behavior, and the inherited biochemical machinery that gives this cultivar its satsats identity. It might bend your brain a bit, but that's part of the fun, because once you understand this level of detail, you stop repeating what you've heard, and you start recognizing what's real. Welcome to Canna Intel 420, where we decode the chemistry. Let's get into it. Durban Poison is frequently spoken of as though it is a singular, unchanging entity with one identity, one composition, and one defined chemical fingerprint. In reality, the name does not point to a rigid formula. It points to a recurring cluster of measurements that appear across independent laboratory analyses, each one capturing a moment in the chemical life of a plant that carries this label. When viewed through a scientific lens, Durban Poison behaves less like a static object and more like a pattern that reappears with enough consistency to be recognized, but never with absolute uniformity. This distinction matters because it determines how the rest of the analysis must be structured. A fixed chemical entity allows for definitive statements. A recurring pattern requires interpretation, comparison, and transparency about where data ends and inference begins. Across the modern era of analytical testing, samples identified as Durban Poison have repeatedly shown similar cannabinoid relationships and terpene structures, but not identical ones. The presence of slight variation does not invalidate the identity. It reveals the biological and agricultural reality that plants express chemistry in gradients rather than absolutes. Seed propagation, environmental influence, nutrient inputs, harvest timing, curing conditions, regional selection, and breeder-specific choices all create subtle drift. What remains stable enough to define is the shape of the pattern, not its exact numerical values. This is why the concept of Durban Poison must be treated as a Chemovar category rather than a single exact chemical recipe. So, every piece of information presented about Durban Poison needs to be placed into one of two zones. The first zone is the factual zone, numbers, relationships, and consistencies that appear across validated laboratory measurements with a repeatability strong enough to stand on their own. Anything placed in this category is grounded directly in documented chemical analysis. The second zone is the speculative zone, details that emerge from recurring tendencies, but which cannot be declared universally true for every expression of the chemovar. When patterns repeat strongly enough, they deserve discussion, but they must be identified as patterns, not certainties. This separation between factual and speculative is not a stylistic choice. It is required to keep the analysis scientifically defensible. Cannabis nomenclature has historically blended storytelling, regional tradition, phenotype memory, and interpersonal lineage claims. A chemical analysis cannot operate in that space. It has to strip away every narrative or cultural attachment and focus only on measurable properties. That means every claim must be supported by something that has been physically tested, recorded, and reproducible. Any point that depends on inference, even if the inference is highly probable and widely accepted, must be labeled clearly to prevent it from being mistaken as scientific fact. The scientific handling of Durban poison therefore begins with acknowledging that the name refers to a cluster of chemical behaviors rather than a single formula. This also clarifies why laboratory results sometimes show variations in dominant terpenes, cannabinoid ratios, or minor compound expression. These deviations do not erase the identity, they illustrate the boundaries of the pattern. The task is not to impose an imaginary uniformity, but to identify which features repeat strongly enough to define the chemovar, and which features drift because the plant is a biological organism subject to change. By establishing the distinction between fixed identity and recurring pattern at the very start, the analytical framework becomes stable. It prevents overreach. It avoids the trap of treating any one sample as the definitive expression. It anchors every claim in measurement rather than mythology. And it ensures that as this deep dive progresses into cannabinoids, terpenes, morphology, and environmental response, each section remains grounded in defensible scientific structure. When examining laboratory analyses associated with samples carrying the Durban poison designation, the most stable and recurring feature is the dominance of tetrahydrocannabinolic acid. This compound exists in the plant as the acidic precursor to tetrahydrocannabinol. It does not deliver the pharmacological effects associated with its neutral form until it undergoes decarboxylation, a heat-driven reaction that removes a carboxyl group and transforms it into the active molecule. Because laboratories typically report both forms, acidic and neutral, the combined total reflects the true cannabinoid potential of the flower once this natural conversion occurs. In the case of Durban poison, that combined value repeatedly falls within a high range, often landing between the upper teens and the mid-20s by percentage of dry weight. This band of concentration does not define a fixed number, but demonstrates a consistent magnitude, reinforcing the chemovar's identity as a high tetrahydrocannabinol expression. The prominence of tetrahydrocannabinolic acid within the plant's resinous structures reflects both genetic inheritance and the biosynthetic priorities of this lineage. In cannabis biochemistry, the plant produces cannabigerolic acid as a universal precursor. Three major pathways compete for this precursor, one leading to tetrahydrocannabinolic acid, one leading to cannabidiolic acid, and one leading to cannabichromenic acid. A plant that consistently channels most of its precursor into the tetrahydrocannabinolic acid pathway displays a genetic bias toward that branch, meaning its enzymatic machinery favors the synthesis of tetrahydrocannabinolic acid synthase over its cannabidiol or cannabichromene counterparts. Durban poison repeatedly shows this bias. This is why its cannabidiol levels remain low, not because cannabidiol is somehow suppressed, but because the biosynthetic competition is resolved very decisively toward one direction. The extremely low expression of cannabidiol found in analyses of Durban poison samples strengthens this interpretation. The cannabidiol pathway is present in all cannabis plants, but the degree to which it is expressed varies widely. And in some lineages, the enzyme responsible for transforming cannabigerolic acid into cannabidiolic acid is minimally active. In the case of Durban poison, the cannabidiol output is typically so small that its presence registers only as trace amounts. This is a hallmark of chemovars where tetrahydrocannabinolic acid synthesis is genetically dominant. The low cannabidiol values, therefore, do not represent an absence, but a clear imbalance in biochemical priority. Cannabigerol, although low in concentration compared to tetrahydrocannabinolic acid, appears consistently enough to be considered a stable part of Durban poison's chemical baseline. Cannabigerol exists chiefly in its acidic form within raw flower, and its presence provides insight into the efficiency of the plant's enzymatic conversion processes. When a plant converts nearly all of its cannabigerolic acid into downstream products, cannabigerol remains extremely low. When a plant converts inefficiently or variably, cannabigerol appears at detectable levels. The values measured in Durban poison show that while most of the precursor is successfully routed into the tetrahydrocannabinolic acid branch, a small but reliably measurable fraction remains unconverted. This creates a repeating minor footprint that, while not chemically defining on its own, contributes to the overall consistency of the chemovar's cannabinoid identity. Tetrahydrocannabivarin is one of the more distinctive recurring components in laboratory data associated with this lineage. It exists as an acidic precursor, just like tetrahydrocannabinolic acid, and undergoes the same decarboxylation process to become active. What separates tetrahydrocannabivarin from other minors is the reliability with which it appears. While many cannabis chemovars express minor cannabinoids inconsistently, tetrahydrocannabivarin shows up with enough frequency and stability in Durban Poison samples to be considered a signature feature rather than a rare occurrence. Its consistent presence suggests a genetic pathway capable of producing varinic cannabinoids alongside the traditional tetrahydrocannabinolic acid pathway, indicating that the plant expresses both the standard synthase enzyme and the variant responsible for producing these shorter chain molecules. This dual expression pattern is less common across modern chemovars, which makes its recurrence in Durban Poison analytically meaningful. Taken together, these components form the cannabinoid backbone that recurs across verified laboratory measurements. The dominant presence of tetrahydrocannabinolic acid establishes the chemovar's primary chemical character. The near absence of cannabidiol reveals the competitive structure of its biosynthetic pathways. The modest but consistent expression of cannabigerol reflects enzymatic efficiency and precursor utilization. The repeating presence of tetrahydrocannabivarin adds a recognizable minor signature that strengthens the coherence of the pattern. When viewed collectively, rather than individually, these features produce a reproducible cannabinoid architecture that appears again and again, allowing it to be recognized even as other variables shift. Among all the measurable features associated with the Durban Poison chemovar, the terpene structure provides one of the most recognizable and persistent patterns. While the cannabinoid profile establishes the chemical backbone, the terpene distribution shapes the sensory and functional identity that researchers repeatedly observe in documented analyses. These terpenes are not random. They follow a recurrent architecture dominated by a specific group of monoterpenes that appear again and again across verified measurements. The two compounds that most consistently anchor this structure are terpinolene and myrcene. In many cannabis chemovars, one terpene tends to dominate, with several supporting compounds forming a secondary layer. In Durban Poison, terpinolene frequently occupies that dominant position, creating a broad monoterpene peak that immediately stands out. Terpinolene's molecular behavior is defined by a light, volatile structure, and its presence in high relative proportions is characteristic of only a small fraction of modern cultivars. When this compound rises to the top of the terpene distribution, it suggests a lineage that carries genetic instructions favoring the synthesis of terpinolene synthase over other terpene pathways. Durban Poison repeatedly demonstrates this preference, often placing terpinolene at the center of its aromatic and biochemical identity. Myrcene forms the second major anchor in this structure. While terpinolene establishes an energetic and bright upper range, myrcene contributes a denser, earthier monoterpene base. The coexistence of these two terpenes in significant proportions is less common across the broader cannabis population, which is one reason the Durban Poison pattern is chemically distinguishable. Myrcene's repeated presence suggests that its synthase pathway remains active even when terpinolene dominates, creating a dual monoterpene architecture rather than a single compound peak. This dual structure also reflects how the plant allocates carbon during terpene biosynthesis. When both terpinolene and myrcene appear strongly, it indicates that the plant's enzymatic machinery is divided between two major monoterpene pathways rather than channeling all resources into a single product. Supporting this foundation is a group of secondary monoterpenes that frequently appear in conjunction with the two dominant anchors. Among the most recurrent is ocimene, known for its volatility and its tendency to degrade during extended drying or curing, which makes its continued detection in Durban Poison samples analytically significant. Its presence suggests that these flowers maintain enough of the fresh, volatile fraction to preserve this compound. When ocimene appears consistently, it reinforces the interpretation that Durban Poison expresses a broad monoterpene suite rather than a narrow or simplified terpene arrangement. Pinene is another supporting terpene that repeatedly appears within this chemovar's structure. Pinene exists in two isomeric forms and often contributes to the sharper, more resinous components of the terpene profile. Its recurring appearance indicates that the terpene synthase pathways responsible for pinene production operate alongside the terpinolene and myrcene pathways. This creates a layered biochemical environment in which multiple monoterpene syntheses remain active within the same genetic lineage, producing a more complex aromatic and chemical footprint than many contemporary high tetrahydrocannabinol cultivars. While this pattern is highly consistent, one documented exception demonstrates the natural variability contained within the chemovar. In a broader metabolomics analysis, a single Durban Poison sample was categorized as myrcene dominant rather than terpinolene dominant. This divergence does not undermine the terpene identity of the chemovar. Instead, it highlights that Durban Poison exists within a narrow expressive range rather than a single, absolute terpene configuration. Biological systems do not produce identical chemical outcomes across all environments, and terpene expression is particularly sensitive to changes in temperature, light intensity, nutrient balance, and curing conditions. When myrcene overtakes terpinolene, it reflects environmental or genetic drift that alters the ratio but leaves the overall monoterpene architecture intact. Taken as a whole, this terpene profile forms a recurrent framework. Terpinolene sets the upper profile with its sharp monoterpene volatility. Myrcene provides the stabilizing secondary anchor. Ocimene and pinene reinforce the lineage by showing that multiple monoterpene pathways remain active in the plant. The occasional shift between terpinolene-dominant and myrcene-dominant expressions simply illustrates the elasticity of the chemovar rather than contradicting its identity. To understand Durban Poison's functional identity in a strictly chemical and scientifically grounded way, the next step is to examine how its dominant compounds interact with the body's endocannabinoid system. This requires separating what is established through biochemical research from what remains unconfirmed. Tetrahydrocannabinol has well-documented receptor activity. Terpinolene, myrcene, ocimene, and pinene do not bind to cannabinoid receptors in the same direct manner, but influence the endocannabinoid system through secondary mechanisms. This page focuses only on scientifically supported interactions without implying medical outcomes or therapeutic claims. Tetrahydrocannabinol binds to the CB1 and CB2 receptors as a partial agonist. This means it does not activate these receptors fully, but instead stimulates them to a measured degree. CB1 receptors are concentrated in the central nervous system, particularly in regions associated with perception, memory, coordination, and reward signaling. When tetrahydrocannabinol binds to CB1 receptors, it alters the release of neurotransmitters by decreasing the efficiency of presynaptic calcium channels and increasing potassium channel activity. This shift changes the probability of neurotransmitter release, producing the electrical modulation that researchers have described for decades. CB2 receptors are found primarily in immune-related tissues and peripheral systems. Tetrahydrocannabinol's binding to CB2 is weaker than its binding to CB1, but the interaction still occurs and influences cellular signaling in ways that are chemically measurable even when subjective effects are less pronounced. The terpenes dominant in Durban Poison do not bind to CB1 or CB2 receptors with the same direct affinity as tetrahydrocannabinol. Instead, they modulate the broader endocannabinoid environment by affecting membrane fluidity, ion channels, neurotransmitter behavior, and receptor conformation. Myrcene has been shown to influence cell membrane permeability, which can alter how fast certain molecules pass into or across cellular layers. Although this does not change CB1 or CB2 receptor activity, By direct binding, it can change the availability of tetrahydrocannabinol at the receptor site by adjusting how the body absorbs or distributes lipophilic compounds. This does not amplify the chemical effect of tetrahydrocannabinol in a pharmacological sense, but it may influence the kinetics of how quickly tetrahydrocannabinol reaches certain tissues. Terpinolene interacts differently. It does not demonstrate meaningful affinity for CB1 or CB2 receptors, but it does participate in modulatory actions through its influence on voltage-gated channels and neural excitability. When terpinolene is present at notable concentrations, it contributes to a chemical environment where neuronal firing patterns shift due to changes in oxidative and enzymatic behavior. These changes do not represent cannabinoid receptor activity, but they create a secondary layer of modulation that occurs in parallel to tetrahydrocannabinol's receptor-mediated actions. This makes terpinolene chemically relevant even without direct receptor binding. Ocimene, the monoterpene rendered phonetically for clarity, behaves similarly to terpinolene in that it does not exhibit strong binding to CB1 or CB2 receptors. Its measurable contributions come from its influence on metabolic enzymes, particularly the cytochrome pathways responsible for the breakdown of cannabinoids. When these enzymes slow or accelerate due to terpene presence, they change the metabolic curve of tetrahydrocannabinol indirectly. This is not a receptor-level interaction, but a metabolic one. The significance lies in the fact that tetrahydrocannabinol's activity at CB1 and CB2 receptors depends not only on its binding affinity, but also on how long it remains available before enzymatic degradation reduces its concentration. Pinene adds another layer to this chemical environment. Although pinene does not bind to CB1 or CB2 receptors, it interacts with several neurotransmitter systems that operate in parallel to the endocannabinoid receptors. Pinene has been shown to influence the behavior of acetylcholinesterase, the enzyme responsible for breaking down the neurotransmitter acetylcholine. When this enzyme's activity shifts, it indirectly affects signaling patterns in regions that also contain CB1 receptors. This interaction is not a cannabinoid receptor effect, but a contextual effect that changes the overall signaling landscape in which tetrahydrocannabinol is operating. Because CB1 activity depends on the excitability and neurotransmitter state of nearby neurons, any terpene that shifts parallel neurotransmitter processes becomes part of the broader chemical interplay. None of these terpenes override the core receptor activity of tetrahydrocannabinol. Instead, they create a chemical environment that influences how tetrahydrocannabinol interacts with CB1 and CB2 receptors. They may adjust membrane behavior, neurotransmitter conditions, enzyme activity, or cellular excitability, all of which create secondary layers of modulation. These interactions are measurable and grounded in chemical behavior, not speculative narratives. The end result is a complex biochemical environment where tetrahydrocannabinol remains the only major actor directly binding to cannabinoid receptors, while the dominant terpenes adjust the conditions under which that binding occurs. When discussing how tetrahydrocannabinol and the dominant monoterpenes found in Durban Poison interact with the human body, it is important to frame them within chemistry that humans encounter every single day. The endocannabinoid system does not exist for cannabis. It exists because the human body produces its own cannabinoids and responds to a wide range of plant compounds found throughout the diet. The chemical behaviors described in previous pages fit naturally into this broader biological landscape. Human physiology is built around receptor networks that evolved long before cannabis was ever cultivated. CB1 and CB2 receptors respond to lipid-based signaling molecules the body generates on its own, such as anandamide and 2-arachidonyl glycerol. These internal compounds guide appetite, stress recovery, sleep-wake balance, inflammatory responses, and memory processing. When tetrahydrocannabinol binds to these same receptors, it is not introducing an alien mechanism. It is engaging a system that already operates daily, even in individuals who never consume cannabis. The receptor activity triggered by tetrahydrocannabinol is therefore not outside the normal range of human biology. It is a redirection of a pathway that constantly responds to internal fat-derived messengers from common dietary inputs. The terpenes associated with Durban poison fit even more seamlessly into familiar dietary chemistry. Terpinolene appears naturally in apples, cumin, nutmeg, and certain leafy greens. Myrcene is abundant in thyme, lemongrass, and mangoes. Ocimene occurs in basil, mint, and parsley. Pinene is present in rosemary, sage, and many conifer-derived foods and seasonings. Humans already ingest these terpenes through daily nutrition, meaning the body recognizes and metabolizes them through pathways used for countless other plant-derived compounds. These monoterpenes travel through the digestive or respiratory system and integrate into the same enzymatic networks that process citrus oils, herbal volatiles, and aromatic compounds found in spices and vegetables. Their influence on human physiology emerges from the same biochemical principles that apply to terpene-rich foods. A terpene that changes membrane fluidity or modulates neurotransmitter behavior does so because its molecular structure interacts with lipid layers and enzyme surfaces in a predictable way. These interactions do not require specialized receptors. They operate through basic membrane dynamics, ion channel modulation, and enzyme regulation, all of which occur continuously as the body processes ordinary dietary components. The indirect influence these terpenes exert on the endocannabinoid system fits within this everyday biochemical logic. For example, shifts in the rate at which metabolic enzymes break down. Bromotetrahydrocannabinol tetrahydrocannabinol are conceptually similar to how certain foods alter caffeine metabolism or how spicy compounds influence the breakdown of alcohol. The human liver relies on cytochrome systems that process a wide spectrum of natural compounds, and terpenes enter this same metabolic arena. When one of these monoterpenes slows or accelerates a specific cytochrome activity, it is participating in a mechanism the body uses routinely whenever it encounters plant volatiles from cooking herbs or fruits. Even the relationship between tetrahydrocannabinol and daily dietary fats reflects familiar metabolic pathways. Tetrahydrocannabinol's affinity for lipid structures mirrors how fat-soluble vitamins and omega-derived messengers travel through the human body. The same transport principles that move vitamin E or carotenoids through cellular membranes also govern how tetrahydrocannabinol distributes within tissues. This is why the body handles tetrahydrocannabinol predictably in the context of lipid metabolism, utilizing the same absorption pathways that it uses for dietary fats. Viewed through this lens, the chemistry of Durban poison does not operate outside normal human biology. It interacts with systems that already respond to plant volatiles, dietary lipids, herbal aromatics, and internally produced cannabinoids. Tetrahydrocannabinol engages the body's own receptor networks, while the dominant terpenes participate in the same metabolic and cellular mechanisms involved in processing common foods and spices. These connections place the entire chemical profile of Durban poison within a familiar, everyday physiological framework, rather than isolating it as something exotic or disconnected from normal human function. The digestive phase of this breakdown must distinguish between two entirely different chemical events: the way the human body processes whole, intact plant material containing its original cannabinoid terpene matrix, and the way it processes manufactured edibles built from isolated cannabinoids and added terpenes. These are not variations of the same thing. They are fundamentally different chemical systems moving through the digestive tract along different pathways, triggering different enzymatic sequences, and producing different downstream metabolites. A great deal of confusion in the public comes from treating them as interchangeable. They are not. When the human digestive system receives pure plant material, meaning flour that still contains its original lipid structures, its full monoterpene profile, and its natural acidic cannabinoids, the process begins with the breakdown of the plant matrix itself. The stomach and small intestine do not just encounter tetrahydrocannabinol. They encounter fibers, waxes, plant lipids, chlorophylls, and the volatile terpenes embedded within that matrix. These components determine how quickly cannabinoids dissolve, how they bind to fat micelles, and how they enter the lymphatic system. The acidic forms, such as tetrahydrocannabinolic acid, do not become active until they are exposed to heat. If the plant is consumed raw or minimally heated, much of the chemistry remains in the acidic state and behaves differently during digestion. In other words, the digestive experience of whole plant material is shaped by the plant's original structure, not by isolated compounds. The natural monoterpenes present in Durban Poison, terpinolene, myrcene, ocimene, and pinene remain embedded in the plant's lipid environment when consumed. These terpenes are highly volatile when exposed to air or heat, but when ingested inside the plant's natural resinous matrix, they are carried through digestion as part of a complex, fat-based mixture. Their absorption begins in the small intestine, where monoterpenes dissolve into dietary fats and enter the body through the same channels that handle omega oils, fat-soluble vitamins, and plant-derived lipids. During this process, the terpenes interact with metabolic enzymes at the same time that tetrahydrocannabinol is undergoing first-pass metabolism. Because these terpenes originate from the intact plant matrix, their influence on metabolic speed, enzyme activity, and membrane behavior occurs in synchrony with the cannabinoids. This is not something recreated by combining isolated ingredients. When a person consumes a stripped-down edible, such as a gummy containing isolated tetrahydrocannabinol infused with terpenes added after extraction, the digestive interaction is fundamentally different. The body is not encountering a unified chemical system, it is encountering two separate chemical streams that have been manually combined. The isolated tetrahydrocannabinol travels through digestion as a purified compound dissolved in carrier fats. The added terpenes, however, are not bound inside the plant's natural lipid structures. They are floating freely in the edible medium, often dissolved in artificial carriers or ethanol-derived solvents. When digestion begins, the terpenes and cannabinoids enter the metabolic pathways separately, not as a synchronized matrix the way they would inside a whole flower. In pure plant form, the terpenes and cannabinoids share the same micelles as they travel through the intestine. Their relative ratios remain intact from the moment they leave the plant tissue until the moment they cross into circulation. This shared passage influences the kinetics of absorption. It shapes how quickly tetrahydrocannabinol reaches liver enzymes, how the terpenes interact with cytochrome systems, and how the compounds compete or cooperate for metabolic attention. Because this co-traveling arrangement is genetically dictated by the plant, it produces predictable patterns, especially in chemovars like Durban Poison, where the monoterpene architecture is stable. The digestion of this intact matrix reflects the plant's original chemical priorities. In contrast, edibles built from isolated tetrahydrocannabinol and added terpenes do not preserve this relationship. The terpenes may be metabolized earlier, later, or unevenly, depending on the formulation. They may partition differently into fat micelles. They may evaporate during manufacturing. They may fragment or oxidize during processing. The result is that the body does not receive the chemical system in the proportions or structural relationships the plant originally produced. Even if the formulation attempts to mimic the terpene profile of Durban Poison, the absence of the plant's natural lipid matrix means the digestive chemistry will not mirror the intact flower's behavior. There is also a structural difference in how the liver processes these two forms. Whole plant material presents cannabinoids and terpenes to the liver in a staggered, matrix-controlled manner, because the compounds are released gradually as the plant tissue breaks down. Isolated tetrahydrocannabinol in a gummy arrives as a sudden purified dose, with terpenes entering the system separately. This changes how first-pass metabolism operates. The liver may convert isolated tetrahydrocannabinol into its metabolites at a different rate than it would when processing cannabinoids embedded in a complex plant matrix alongside natural monoterpenes. Because the terpenes modulate enzyme behavior, their timing of arrival matters. When terpenes arrive out of sync with cannabinoids, their modulatory influence shifts. This is the core distinction. The original genetic chemistry of the plant creates a unified biochemical system that digestion recognizes and processes as a single matrix. Stripped-down edibles with added terpenes are composites of isolated chemicals that digestion treats separately, altering absorption, metabolism, and overall chemical behavior. When examining how pure flower behaves through digestion, the final step is understanding why that behavior is so consistent across different samples of Durban poison in the first place. The answer sits in lineage chemistry. The digestive system responds predictably to Durban poison because the plant itself produces a predictable internal chemical structure, one that remains stable across generations, regions, and environmental variation. Lineage chemistry explains how the plant's genetic code dictates the composition of the cannabinoid terpene matrix that digestion later interacts with. The most foundational lineage trait visible in Durban poison is its enzyme hierarchy. The plant reliably prioritizes the conversion of cannabigerolic acid into tetrahydrocannabinolic acid rather than into cannabidiolic acid or cannabichromenic acid. This is why its cannabinoid backbone remains so consistent. It is not the result of cultivation technique or regional influence. It originates in the structure of the genes responsible for cannabinoid synthesis, which direct the plant toward high tetrahydrocannabinolic acid production regardless of external conditions. The digestive distinctions in how the body encounters the full matrix of acidic and neutral cannabinoids depend directly on this inherited enzyme bias. Alongside this primary pathway sits another inherited feature, the varinic branch of cannabinoid synthesis. Durban poison regularly produces tetrahydrocannabivarin, a 3-carbon chain cannabinoid that requires a separate enzymatic route from the standard tetrahydrocannabinolic acid pathway. Many modern cultivars rarely express varinic cannabinoids at meaningful levels, making the repeated appearance of tetrahydrocannabivarin in Durban poison a genetic signature rather than a byproduct of environment. This varinic expression becomes part of the intact biochemical matrix that digestion processes as a unified system, linking its presence not to cultivation style, but to inherited molecular architecture. This same inheritance pattern appears just as clearly in its terpene system. The digestive differences between whole plant chemistry and manufactured edibles only matter because the original terpene profile behaves consistently from one generation to the next. Durban poison is genetically inclined to produce a monoterpene-heavy architecture anchored by terpinolene and myrcene. Even when environmental variables shift terpene ratios slightly, the plant does not drift into lanolule-dominant or humulene-dominant territory. It remains within a narrow terpene expression band because the genes controlling monoterpene synthase pathways are stable. This reliability is what forms the terpene component of the digestive matrix and is why whole flower delivers terpene-cannabinoid relationships that manufactured edibles cannot replicate. Lineage chemistry also governs how tightly these pathways are linked inside the plant. Durban poison does not express its dominant cannabinoids and terpenes as isolated outputs. Its genetic code drives them to form together within the same resinous structures, creating a naturally unified lipid terpene cannabinoid environment. This inherited structural cohesion is the reason digestion receives these compounds as a single integrated system rather than as separate chemicals. The gastrointestinal tract responds to that matrix as a complete biochemical unit because the plant's lineage ensures it is produced as a complete biochemical unit. Durban poison's lineage chemistry therefore functions as the bridge between its internal biochemical construction and its external behavior inside the human digestive system. The consistent enzyme pathways, the varinic expression, the monoterpene architecture, and the unified resin matrix are not environmental accidents. They are inherited genetic directives. These directives shape how the plant builds its chemistry, and in turn, how the human body processes that chemistry when the plant is consumed in its original form. The relationships between cannabinoids, dominant monoterpenes, receptor activity, physiological familiarity, digestive behavior, and inherited genetic pathways now form a complete biochemical picture. Each element connects to the next through measurable chemistry rather than assumption, and the structure holds together without needing anything added to it. The foundation is fully built, the internal coherence is established, and the lineage-driven architecture that shapes this entire profile stands clear on its own. Nothing further belongs in this segment. The next movement begins only when we shift into a new angle of analysis, and that, my friends, is the moment where your brain officially deserves a nap, a snack, or possibly a small emotional support pillow. We just walked through the chemical architecture of Durban Poison like it was a college final nobody warned you about. If you made it this far, you're basically an honorary biochemist now, just without the student loans or the questionable haircut choices that usually come with that profession. This deep dive wasn't just about numbers on a lab sheet. It was about understanding how this plant actually builds itself, how your body recognizes that chemistry, how digestion handles the real thing versus the lab-built version, and how the lineage holds the entire system together. If your head feels warm, that means you understood it. If your eyes glazed over somewhere around enzyme hierarchy, don't worry, that's normal. Even enzymes get tired. But here's the thing. This is the kind of knowledge that actually matters. This is the difference between repeating hype and understanding the chemistry beneath it. And that's why we do this work. That's why Cannabinoid 420 exists. To take the complicated stuff, break it open, and show you how it actually functions without the fluff, the mythology, or the recycled talking points. I'm Jerry, and if you stuck with me through all of this, through the cannabinoids, through the terpenes, through the receptor interactions, through the digestive matrix, through the lineage, you're exactly the kind of person who belongs here. People who want the real story, told straight, built on data, and delivered with a little bit of fire. So take a breath, hydrate, let your brain cool down, and remember, at Cannabinoid 420, we don't chase trends. We break them open and show you what's inside. Thanks for being here. Thanks for learning with me. And as always, stay curious, stay analytical, and stay rooted. I'll see you in the next one.